the return of life to our yard was very important and revealed sort of fundamental truths to us about the role of microbial life in supporting the life we know above ground. But along the way, we came in for another surprise. About the time that we were really convinced of the role of microbial life in supporting plants in our yard, Anne got a terrible surprise. She was diagnosed with cancer. Fortunately, it's behind her. She was diagnosed early and treated very effectively, and now several years later on, she's doing okay. But much to our surprise, it was a microbially generated cancer. There are some cancers that are caused by viruses. She had one that was probably rooted in a virus. And this got us really thinking about, well, what's the role? I mean, a cancer diagnosis motivates you to think about your health. It motivates you to think about the human immune system. It motivates you to rethink your diet. All these things went into a mix in our lives when it got turned upside down by this out-of-the-blue diagnosis. But it also made us start looking into what's the role of microbial life in human health. We know about sort of colds and the downside, but at about the same time that we're going through this, the Human Microbiome Project was essentially revealing that the genetic diversity in our microbial life greatly outweighs the diversity in our own genes. That the genes in the human microbiome outnumber those in the human genome by about 700 to 1 or something. Those numbers probably aren't very precise, but the point is that we started to realize just how much of our own bodies is actually integrated with and related to the microbial life within our bodies. And there's been an explosion of microbiome research over the last couple decades, and the last decade in particular, that's really revealed strong connections between microbial life, soil fertility, and human health. And as we started looking into that literature, we started to really see microbes in a whole different way in terms of the way they interact with our own health. In particular, we were surprised to discover that the human body has within it something like 100 trillion microbes, most of which live in our colon. Both of those facts to us were quite surprising. The idea that there were as many microbes living within us as there are stars in the galaxy in which we live was really kind of mind-stretching. But the idea that most of those microbes were living in the colon was really, that was kind of a surprise. We hadn't thought much about our colons, to be frank, and quite happily so, I guess, for many years. But we were also surprised to discover, in looking into the human immune system and what we could do to bolster the immune system, in part because of Anne's diagnosis. We were surprised to discover that 80% of the human immune system, or human immune cells, is associated with the gut, with the colon in particular. And that figure that shows you that, it's up on the screen now, sort of shows you the cutaway blow-up of the human colon, and that if you look at the big slice there, you notice that there's a few things called out. There's a mucus layer that you have, and there's a thing called the galt and the colon crypt, so I'll sort of go through what each of those are. That mucus is essentially mucus that the goblet cells produce, that it's a sugary, it's a carbohydrate-rich substance that bacteria in your colon wall and lining eat. Now, your colon is not just exuding mucus to feed those microbes, it also keeps things moving along in a way you'd like to keep them moving along through that particular piece of anatomy. But it also feeds microbes. And we started thinking, wow, is that what plants are doing? Is that like an exudate? And it turns out that that thing called the galt over there is the gut-associated lymphatic tissue, is that part of the colon in which an awful lot of your immune cells reside. So a lot of your immune cells are sort of sitting just on the other side of that mucosal lining of the colon. And if you look down into the colon crypts, there's still a lot of research that's going on this in terms of sort of what's living in there in terms of microbes that are in there. But the thinking is that there are some microbes that are actually inhabiting these deep recessions in the colon wall, those vertical crypts that if you look at in a micrograph, look a lot like this, where on the left what you see is essentially a healthy colon lining with divided into little segments of which there's a pit right down in the middle. And you'd have to be very small to fall into those pits. You'll notice that the scale bar across the bottom there is 1/10 of a millimeter. Now, if you're a microbe, that's still a huge hole. You can like drop right down it. But if you're a millimeter-sized person, you could sort of span it with a leg span. And if you take each one of those crypts and you sort of pull it out and isolate it, what you find is it's essentially a tube. It's a little gland that is essentially a singular tube, and they're all stuck together to form the lining of the colon. And what do? Well, I think that we're still discovering, I think microbiologists, again, Ann and I are a geologist and a biologist, so we're not discovering any of this stuff. We're just reading other people's stuff and learning it. But what I think is still being discovered is essentially what role do those crypts play and which microbes are actually living in them. But it's a reasonable idea to think that they're not there to support pathogens. There are areas where your body is actually exuding carbohydrates in what could be ideal microbial habitat. Odds are, I think, that there's something else going on. And if we think about what that might be, if you think back to that GALT, that gut-associated lymphatic tissue that's just on the other side of the lining of the colon, it's populated by a lot of immune cells, in particular dendritic cells. And what do? Well, they collect antigen. And what's antigen? Well, it's a microbial sample of essentially, it's like a piece of a microbe. You could think of it as taking a little piece out of a microbe. And dendritic cells are very odd cells. They're kind of like, everybody remember the movie The Blob, where it can like ooze out in different directions, sort of like a giant amoeba. Well, dendritic cells are like that. They're the one cell that actually can extend an arm through the lining of your colon, squeezing it in between cells, and it goes up into the lumen, which we call the interior of your colon, and into the mucosal lining of the colon. And actually, it extends it out like a periscope, but it's a periscope with a grabber on the end, and it can actually grab a sample of whatever microbes are living in those environments and take that antigen, that microbial sample, and pull it back down into your body. Well, why would it do that? That's a fairly strange thing for some cell to do. Well, it turns out that it does that to go show it to your other immune cells. You could think of your dendritic cells as prospectors that are going out and actually looking for what is actually in your gut. And it takes that antigen sample and it brings it back to show to other immune cells, in particular T cells. And T cells are an interesting kind of cell that has a particular receptors that are on it. That's sort of shown in that slide as a T cell receptor. And each T cell receptor is unique. It's matched. It's kind of like a hand that has a perfect counterpart, and that to give you the perfect handshake or the lines up, or the identical to mirror image hand. I'm not sure what the best analogy is, but you get the point. It's a unique. It's a unique receptor that matches a particular compound. And so when a dendritic cell brings back an antigen that does not match that compound, the T cell kind of ignores it. When the T cell brings back an antigen that is perfectly fit to its receptor, it activates. It basically turns into the kind of T cell that I'll be talking about. Well, it basically does things. And so the dendritic cells are prospecting for the microbes that are living in your colon, bringing samples back in, showing it to other cells in your immune system, and what do they do with that? Well, T cells are activated by the antigen the dendritic cells bring back, and what T cells are, they're immune cells that either promote or quell inflammation. And they also kill infected cells. But there's different kinds of T cells. Some of them promote inflammation, some of them combat inflammation or quell inflammation. And as was mentioned on the panel last night, inflammation is not necessarily a bad thing. You need it to heal. If you cut your finger, you want inflammation to come in and start helping to repair those tissues. But too much inflammation, chronic inflammation in particular, is incredibly bad and can lead to many of the kinds of chronic illnesses that you've probably been talking about this week. And it turns out that T cells act in a way as a regulatory system that can dial that up and dial that down. And if you look at the image on the right, it basically illustrates how there's different kinds of dendritic cells sampling different kinds of microbes and bring back different kinds of antigen to your immune system can activate different kinds of T cells. Some of the T cells, like the Tregs shown there on the right, serve anti-inflammatory purposes. They essentially can, if your dendritic cells are bringing back antigen that is activating T cells that quell inflammation, it's going to be putting a damper on inflammation. On the other hand, if your T cells, like the one on the left, are bringing back antigen that activate Th17 cells that are pro-inflammatory, then essentially inflammation can be kicked on and amplified. In other words, you could think of your T cells in terms of which ones are being activated as a teeter-totter balance that essentially is intended to maintain an optimum level. Well, optimum is kind of the wrong motivation to put on a system like this, but that would maintain a balance of inflammation, calling it in when you need it and quelling it when you don't, essentially maintaining a healthy balance of inflammation in your body. And the realization that microbes communicate the information essential for the smooth operation of the immune system through that dendritic cell sampling of antigen and activation of different kinds of T cells, the idea that those microbes are essential for controlling the level of inflammation in your body gave to Ann and I, at least, when we sort of looked into the research behind this, sort of a whole new way of looking at microbes in our body. Maybe they're actually essential for health in ways that we hadn't thought about. And why hadn't we thought about that? Well, like probably just about everyone else in America, we've been steeped in germ theory for most of our lives. The idea that microbes are germs, that they're bad, that they're out to get you, they could harm you, they could kill you. This is thinking that goes back to the 19th century when it was well discovered that particular microbes were indeed responsible for particular diseases. And if you look back at the causes of mortality back, say, in the 17th century, this may be a bit blurry to read there, but if you go through and read sort of all those causes of mortality from one of the bills of mortality in London in the 17th century, you'll notice that an awful lot of those things that took people out were microbially generated: fevers, plague. There's other things like the King's Evil. I don't know what that was, but only one person died of it, apparently. You look at the big numbers on there, and it's essentially microbial stuff. There's no doubt that many microbes were pathogens. There's something like 1,400 different human pathogens, but there's millions of different kinds of microbes in the human microbiome. Smallpox and polio in particular are two examples of things that, of microbial maladies that afflicted both the rich and poor alike until the development of vaccines. We go into sort of the history of that kind of thinking in the book. I won't go into that today in the interest of time, but over the course of the 20th century, we saw a very large change in the causes of death in the United States. Back in 1900, infectious diseases were taking out about 53% of the people who died. Chronic diseases accounted for just about a third, 36% or so. By 2011, that had really flipped. Infectious diseases were down to about 3% of deaths, and chronic diseases were related to 88%, almost 90%. Of the people who died it was associated with chronic diseases. Now, of course, there's a number of explanations for this, not the least of which is the development and wide use of antibiotics in the 20th century really helped reduce the rates of infectious diseases. Medical practice advanced greatly as well. Sanitation and public health advanced greatly. There's a lot of reasons why the deaths from infectious diseases went down over the 20th century. What that doesn't explain, though, is what about the rise of chronic diseases in the latter half of the 20th century? This graph shows essentially the decline of infectious diseases over on the left-hand side between 1950 and 2000, or they're all sort of going down for many of the reasons that I was just talking about. But on the right-hand side, it's the incidence of chronic diseases. They're rising from the 1950s to the 2000s. Different ones rose at different paces, and you could throw some more on here if you went out and found the studies. But the question is, what actually sort of led to the rise of chronic diseases? And again, I'm a geologist, so don't take any particular medical advice out of this talk at all, other than with the weighting of, yeah, it's from a geologist and a biologist. But one of the things that happened in the 20th century, of course, that we all know about is the rise of antibiotic resistance. The use of antibiotics actually changed not only the microflora within our bodies, but it changed them in they adapted to it. It took about, what each of those bars show is the time between the introduction of an antibiotic onto the market and the first reported rise of incidences of antibiotic resistance. And you notice we've gone through a fair number of antibiotics since the first introduction of penicillin in the 1940s, but you look at the time scales and you're talking a couple years to a couple decades. In other words, it doesn't take microbes all that long to adapt to the new ways that we developed for killing them. And it turns out that that's rooted at some level in two things. One, they have an incredibly short generational turnover time, on the order of 20 minutes. So as I've been talking, they've turned over a few times. And they also have a lot of genetic plasticity. It turns out they can actually trade genes the way we trade stories and handshakes. They don't have to reproduce to modify themselves genetically. It's called horizontal gene transfer. And if you take essentially those two things, something with the ability to share genetic information and with an incredibly short generational turnover time, the idea that we will win a genetic arms race with them starts to sound kind of silly. And that's basically what's been happening with antibiotic resistance, and it's one of the main reasons why the over-application of antibiotics in both medicine and agriculture is not in society's best interest. Now, it can be in our own personal best interest if we need them for a particular malady at the moment, but it's in all our best interest to preserve their ability to work for future generations as well. Well, so the rise of antibiotics and their use and overuse in medicine and agriculture is probably one of the things that was going on. And another one is essentially the changing nature of the diet in the Western world, and particularly in North America, over the course of the 20th century. And this is our sort of attempt to capture that in a single graph, where it shows you carbohydrate consumption from 1910 to 1997, and the way that that's actually changed. So if you look at the early part of the century, we tended to eat a high-carb, high-fiber diet. It was rich in whole grains, unprocessed foods, and over the course, right about the time of the Second World War, from meat consumption, protein consumption went up. We started to eat a lower carb, low fiber diet. And most recently, since the 1970s, 80s, you can put the line where you want to put it, we've essentially transitioned to what is for most people a high carb, low fiber diet. In the sense that we're getting a lot of calories from simple carbohydrates and sugars that we were not consuming in the same quantity early on, and we're not eating anywhere near as much dietary fiber as we used to. Why would this kind of change actually matter to our microbiome and to our own health? Well, to look at that, we ought to look at essentially the human digestive tract as a series of, as a system or as a sequence of ecosystems. And if we look at that, we can essentially divide our digestive tract into the stomach, the small intestine, and the colon. And if you look at the life that's living within there, in the stomach, in terms of bacteria per milliliter, there's not many. There's that one that was mentioned last night, Helicobacter pylori, that can tolerate the acid of the stomach. But the stomach's job in the digestive system is to dissolve things. There's not a lot of life that's living in there. The small intestine, you get a lot more microbial life in it. Its job is to absorb things that were dissolved in the stomach. That's where a lot of the products of the digestion of simple sugars and proteins comes out. And simple sugars are what we tend to call a sugar, simple carbohydrates, short-chain carbohydrates, tend to come out in the small intestine and are absorbed right into the bloodstream. It's the colon that has most of the life in the digestive tract. 10 to the 11th bacteria per milliliter. That's a lot. Anything with 11 zeros after it is a lot. And what happens in the colon? Well, it turns out that you can think of it as a fermentation tank. A lot of the materials that we don't digest, that don't get dissolved in the stomach or absorbed in the small intestine, end up in the colon. And that's the stuff that essentially feeds the microbial life in the colon. And what is it that the microbial life in the colon is essentially adapted to process? What do they live on? Well, that's where that fiber part of the diet comes in. Because we can't break down dietary fiber ourselves. We need microbial assistance to do that. And this is a colored micrograph of bacteria breaking down a piece of fiber. Your gut microflora in the colon, love to digest fiber. That's a key part of what they consume. And it turns out that that's a key part of why their consumption generates things that help you in terms of your health. This graph from a recent paper, not one of ours, shows essentially, is a very nice illustration that shows how that process works. If you start up in the upper left-hand corner, we're going to sort of go around clockwise on the figure. Fiber that makes it all the way to your colon, dietary fiber, the stuff that's indigestible to you with your own genome, whether it's starch or cellulose, plant, and what is that stuff? It's plant matter. It's plant foods. That stuff your microbiota breaks down and transforms it into fatty acid metabolites, things like acetate, butyrate, and propionate, that serve functions within your colon, and among others, keeping it from becoming leaky. But those fatty acid metabolites essentially activate receptors on T cells that quell inflammation. So if you track and follow that around, if you're eating enough fiber to support the microbiota that's generating enough of those fatty acids to trigger the T cells that are Tregs that generate IL-10 or interleukin-10 that acts as an inflammatory response blocker, that's a way to essentially maintain the health and integrity of your colon lining. In other words, it's the things that the microbes in your gut are breaking down and turning them into that your body is using to maintain your health. Now again, I'm a rock doctor, I'm not a medical doctor, so I'm not going to give you any particular advice on any of these particular maladies. But I can read the scientific literature. Anne's a biologist, she's worked in public health. We read this together and essentially look at, there's a large list of maladies that researchers have started to associate with an altered microbiome. In terms of which of the connections to which of these particular maladies is causal and which is just associative, I think that researchers are still working a lot of that out, and we're the wrong people to give you specific advice on any of those. And yet, even if we were, our understanding of the community composition of the microbes in our gut is in its infancy. We're just starting, researchers are just starting to figure out who's living where, like who's living in those crypts in the colon? What are they doing there? How important is it that there's particular species or particular communities of species? And how do you change those? How do you alter them? How do you support their activity and health? We're still at the days of really just discovering that this stuff matters. And that's why we wrote the book in terms of looking at the hidden half of nature, because we think that if you look out 20, 30, 40 years, the worlds of medicine and agriculture are going to be different because of the way people are discovering now how these microbial connections work. So if you look, I'll just give you a couple examples. One, again, taken from the literature, the difference between a symbiosis, or sort of a healthy relationship with your microbiome, and dysbiosis, an unhealthy one, is captured in these two slides of looking at Clostridium difficile, C. difficile, that can create chronic diarrhea and be a major problem for some people. In a healthy gut, C. difficile is present in small numbers, and it's the diversity and abundance of other microbes that keep it in check. It's the community composition that keeps it in check. You could think about that sort of the same way that what happened to deer in North America. As long as there were a lot of predators for the deer around, the deer population was kept under control. And what happened when we took the wolves and the coyotes and the things, well, do coyotes go for deer? The predators that actually took deer out in the past, when you take them out, the deer population explodes and they can become actually a nuisance. Well, the same thing can happen with microbes, because after antibiotic treatments, C. difficile can, if you wipe out your normal gut flora, and if it's the C. difficile that recolonizes first, it can actually lead to long-term problems and infections that over the course of the last couple decades have actually been growing quite dramatically in terms of the numbers of deaths per million people. It's a small number of people affected, but the antibiotic treatments haven't been working too well for this. And there's been many untreatable C. difficile infections. What has actually worked? Fecal transplants. The idea of actually transplanting fecal matter from a person with a healthy microbiome into someone with C. difficile infection, it's actually resulted in something like 90% cure rates in clinical trials. In fact, some clinical trials were terminated early because most of the patients receiving the treatment were being cured, whereas the ones that were not receiving the treatment were continuing to have major problems. What this illustrates, I think, is the power of thinking in terms of microbial applications as medical applications, and how to manipulate our own microbiome in ways that actually enhance our practices, both in medicine, as I'll talk a bit more about in a minute, in terms of agriculture. So what does this stuff all really mean for us? Well, what Ann and I came to realize when we started looking at these parallels between the world of the microbiome in soil and the world of the microbiome in the human gut is that you can really think of the root and the gut as the same kind of system inside out. And of course there's differences. We're talking about plants and people here, right? There's differences. But if you think at the sort of the 30,000 foot level in terms of what's going on in terms of our relationships with our microbes and our microbiome, if you look at roots, the exudates feed the beneficial microbes. In the gut, mucus is feeding beneficial microbes, we think, on the gut lining. In the roots, microbes acquire nutrients for plants and trade them with plants. They also make critical metabolites that they trade with plants. And it turns out that microbes in your gut are also making critical metabolites that help promote your health. Microbes are a key element in communication and plant defense. They're also a key element in communication and teeing up your immune system response, your own defense system. And if you think of the root in plants as, you can think of it as an external digestive system for the plants. It's an external rumen. You know, a cow has a rumen that is a fermentation tank that breaks down organic matter, the grass that it eats. Cows don't actually digest the grass, the microbes in their rumen do. You can think of the rhizosphere as an external rumen for plants. It's an external digestive system that takes that organic matter. All that organic matter Ann was adding to our yard was being digested outside the plant roots and then taken up. It's an external stomach. In us, it's the colon that serves that purpose. We take the plant matter inside us, where the fiber is fermented to make those critical metabolites. So if we look at the parallels between the root system and the gut system, we actually start realizing, oh, maybe one of the fundamental organizing principles of health in both plants and people is constructive, healthy symbioses with the microbial life that we didn't even know about until a couple centuries ago, and that we didn't even think had these kind of effects until researchers started putting this story together over the last couple decades. Well, what does this mean? Well, we're still in the early days of understanding all this stuff, but I think already some key implications are clear from this new perspective. You know, we're not going to be able to rely on broad-spectrum biocides, things that sort of blanketly kill microbes indiscriminately, as our go-to solutions for problems in agriculture and medicine, if most of the microbes out there are actually either beneficial to us or do no harm. If we tailor our strategy to take out just a couple, you know, the 1,400 or so pathogens out of the millions of different kinds of microbes that surround us and are in us, you know, that's really using the wrong tool for the job, because we're taking out a lot of our allies at the same time that we're taking out our enemies. Now, this is not to belittle the progress that antibiotics made in the 20th century at curing infectious diseases. I'm happy that that we have those tools in our quiver. But I'd like to see those tools preserved to be able to help benefit future generations as well, and that the practice of the application of broad-spectrum biocides will forego that as microbes adapt. We need different strategies.